The last part of DNA and protein synthesis we will talk about for Science 30 is protein synthesis. DNA is present in all living things, except for some viruses, although viruses are not technically living until they invade cells. It's responsible for heredity, and it controls protein synthesis. What do proteins do? Enzymes speed up chemical reactions. Hormones and antibodies work to send signals in your body or to fight off invaders. Hemoglobin carries oxygen to blood cells. Collagen is a strength protein. There are receptor molecules in various parts of your body that respond to signals, as well as carrier molecules that move things from one place to another. Keratin forms hard parts of reptiles, birds, amphibians, and mammals. There are also gene regulation and repressor proteins. You are made of billions of cells, but they carry out different functions, regardless of the fact that they have the same identical DNA in each one of their nuclei. How do your pancreatic cells know to make insulin and your skin cells know to make collagen? They are regulated by specific repressor proteins that bind to DNA segments and turn DNA on and off. What are proteins made of? They're made of 20 amino acids. The sequence of amino acids identifies a protein or a polypeptide. The sequence of bases in DNA determines its sequence of amino acids. One gene is equal to one protein. A polypeptide chain of amino acids is linked together by peptide bonds. Each amino acid is coded for 3 nitrogen bases on a DNA molecule. Each 3 nitrogen base is called a triplet. For example, the DNA triplet AAA codes for the amino acid lysine. We can use page 13 of our data books to look up each DNA triplet. It's all there for you. And take a moment and look up the following amino acid triplets: TGC, or TGT, CAT, ATG, and TGA. Polypeptide chains are made of many amino acids arranged in a specific order and linked by peptide bonds. Proteins are large molecules made up of one or more polypeptide chain folded and coiled into a specific shape. So here are some amino acids. The circles represent amino acids. When they're joined together, they're called a polypeptide. When they're folded, they're called a protein. Proteins are classified into 4 types based on the arrangement of amino acids. We first talk about primary structure. Every different protein has a different sequence, and number of amino acids is determined by the genes on the chromosome. Amino acids are organized in a linear or a straight line. So here is a linear primary structure. A secondary structure, amino acid chains are coiled or folded due to hydrogen bonding between the positive and negative ends of the molecule. So you can start getting helix formation or sheet formation. Then you can have a tertiary structure. The coiled or sheet molecule is further twisted and folded into a more three-dimensional shape. A quaternary structure is interactions between different kinds or different protein shapes to form proteins that are made up of more than one polypeptide chain. Examples of these are hemoglobin and different kinds of enzymes.